Okay, good morning everyone. It's great to see so many of you here in our first EM course in the century of LMB. So I think it's very exciting times for electron microscopy. I think we put together a course that should give you a good introduction to the topics, especially if you don't know too much about it, and hopefully there will be then enough material for you to keep on reading and get deeper into it as deep as you want to. I'm very happy that Tony Crowther will be giving our introduction. He has added a historical perspective on with his view of how this field has evolved over the past decades. So I'm very much looking forward to that. Tony?
Sure. So, as George said, this is indeed a very exciting time in electron microscopy. And I think it's diagrammed here on this introductory slide that George made for all the lectures to kind of tie them together. So you're all beavering away down here doing wet things, making wonderful samples. And then we have this box downstairs on the ground floor. It's like a sort of a TARDIS for Doctor Who. You put your wet sample in here and magically a protein structure comes out at almost atomic resolution. I think that's magic. Anyway, the purpose here is to try and explain a little bit how we got to here. It's been quite a long process, and I'm old enough now to have seen quite a lot of it, I'm afraid. And also to just mention one or two of the background topics that you need to understand the program as it goes on. Lots of things will be explained in much more technical detail. I'm going to be sketching over and just providing a general introduction. So in 1700, around 1700, this was state of the art. Okay, this is one of van Leeuwenhoek's microscopes. Eminent microbiologists, it's just like a single lens magnifying glass. It can magnify up to about 300 times. And with this, for the first time, he visualized bacteria. And you could actually see the shape of the bacteria. There were small bacteria, helical bacteria, rod-shaped bacteria. So in a sense, this was the start of structural microbiology. However, obviously, with a microscope like this, or even with improved light microscopes that we have now, you're still limited in the degree of detail that you can image by the wavelength of light. So that we need an electron microscope where the effective wavelength of the electrons, because we can treat electrons as waves as well as particles, is no longer a limiting factor in terms of the fineness of detail that can be resolved. So we have a source of electrons of some kind, a gun, a field emission gun, or a heated filament. Some condenser lenses here which produce a nice coherent beam at the specimen. The sample holder is inserted here on the side with your specimen grid in here, and then the objective lens and the intermediate and projector lenses produce a magnified image of the specimen here on the viewing screen, which you can then, as in the old days, record on photographic film, which was a very good recording medium, but is now being superseded by CCD cameras and direct electron detectors, as you'll hear a lot more about in the course. So that with this kind of microscope, as opposed to this kind of microscope, the state of the art now is that we're essentially producing atomic resolution structures of biological complexes of reasonable size. And this is a slide that I stole from Werner Kuhlbrandt's recent article in Science, which is called The Resolution Revolution. And indeed it is. This is here a homegrown one, the mitochondrial large subunit from the ribosome by Alexei and colleagues. So I'll come back to this, but this is where we're at now. This is today. This is the moment. And you're very privileged to be part of it, I think. So if we go back, the electron microscope was developed by Ernst Ruska and his colleagues in Germany in the 1930s, the late 1930s, and it didn't become available commercially until after the Second World War. Obviously, this interrupted developments of technical things like instrumentation. But this, I think, is one of the earliest biological electron micrographs taken by Helmut Ruska, that's Ernst Ruska's brother, in 1942. And it shows bacteriophage here, and you can just about see the dark phage heads and the little tails sticking out of the back. If you want to find out more about the very early development of electron microscopy, Ruska's Nobel Prize lecture, he got the Nobel Prize in 1986, is available on the Nobel website, and it's well worth reading, actually. It's really quite interesting. And this picture is taken from that document. So, this really is about the stage that the bacterial pictures that I just showed you with the von Leeuwenhoek microscope is at. You can see there's something there. You can see a bit about its shape. In order to visualize these, because biological materials are low atomic weight materials, they don't scatter electrons very strongly. And therefore, you need to provide a contrasting medium, or you used to need to provide a contrasting medium. So in this case, the phage particles have been stained with a heavy metal salt, which has then been washed away just to leave the uranium or whatever, I don't know what the salt was in this case, attached to the biological materials, and therefore it looks dark. It's strongly scattering. Another way of visualizing biological particles was developed a bit later than this so-called positive staining method, was the shadowing method. And here again, a different kind of short-tailed phage here from a picture by Fraser and Williams in 1953. So this was where the electron microscopy was in the 1950s, as I said, a bit like van Leeuwenhoek in the 1700s, seeing the shape of biological particles but no details. And the first development in specimen preparation and preservation was the development of what's called negative staining. Here, again, a field of various kinds of viruses, and it turns out, because viruses are fairly readily available and they're nice particulate specimens, they're robust, the study of viruses, although it's important in itself, has actually underpinned quite a lot of the developments, the technical developments in electron microscopy at each stage. Viruses have often been used as a test specimen. So in negative staining, as opposed to the positive staining, which I just showed you for the Ruska picture, you put the sample, the virus, onto carbon-coated grid, and then you wash it with a dilute solution of something like uranyl acetate, 2% uranyl acetate, blot off the excess, and then let the last film of it dry, rather than washing it completely away as you do with positive staining. And what happens is that the stain, the black here, dries around the biological particle on the grid and nicely outlines the surface of the particle, so that what you see, as it were, is a hole in the stain when you look at a negatively stained particle. And this is why it's called negative staining. This was a term invented by Sidney Brenner when he was still doing microscopy many, many years ago. And the advantage of this now is that it gives you a robust, stable, high contrast specimen, but it also gives you some details. And you can see that within the virus particles here, or here, this is adenovirus, a picture by Bob Horn and his colleagues, or tobacco mosaic virus here. You can now see that you're not just seeing a shape, but you're actually seeing details within the image of the particle, to the extent that it was possible to start thinking about, well, how do we interpret these images? And this is an early attempt at model building based on the assumption that these virus particles are icosahedral. So with this kind of technique, because you could now see at least molecular details, it was worth thinking about how you might interpret the images quantitatively rather than just by this kind of heuristic model building. And the real start of the quantitative aspects of the field, which you're all going to hear about this week, stems from this paper in 1968 by David de Rosier and Aaron Klug here in the lab. And this really did set the scene for the entire development. It's seldom that one paper is absolutely key in the development of the field, but this is one such. And they say general principles are formulated for the objective reconstruction of a three-dimensional object from a set of electron microscope images. These principles are applied to the calculation of a three-dimensional density map of the tail of bacteriophage T4. And that kind of sums it up. And that is a paper, if you're a student starting up in this, you must go and read that paper. It's absolutely essential. It's the key first paper. So the problem is that when you have an electron microscope image here, such as this image of T4, the depth of focus of the electron microscope is much greater than the thickness of the specimen. So that what you record in this two-dimensional image is a two-dimensional projection of the three-dimensional specimen in the direction of the electron beam. So the problem that you're faced with is to recreate from such an image or set of images a three-dimensional map. Now, pretty obviously, for a general case, if you take one projection of some kind of three-dimensional specimen, then you won't. A feature in the image could come from anywhere through the depth of the projection. So it's clear that you must have at least 2 or 3 projections to get back into 3 dimensions. And the case here is somewhat special because what they looked at was the tail structure of the bacteriophage, and this is helical, so that the subunits that build it are already tilted for you. It's as if the tail has a mini built-in tilt stage. And this they exploited. And they also introduced the idea that the use of a mathematical device called a Fourier Transform, which you're going to hear quite a lot about this week, is really key for understanding this. And Fourier Transforms, they're important for understanding the imaging in the microscope. They're important for something called the projection-section relationship, which I'm going to come to in just a moment. You can use them for finding orientations of particles. When you put particles to different views together, you've got to find their relative orientations. You have to do defocus correction. I'll come on to this later. They're technically important because they give you a very efficient way of doing image alignment and correlation. And they're important also for analysing how well you're doing, how good is your map at any given stage of the processing. So Fourier transforms are really central to a lot of the aspects of understanding and using the microscope and its imaging capabilities. So I'd just like to use 4 picture slides to try and introduce you to Fourier transforms, and Georges tomorrow will be taking you on a bit more of a technical journey. But the first one is, I like, from Max Perutzy's Nobel lecture in 1962, which demonstrates the notion of a Fourier transform in the context of a crystal, because he was talking about X-ray crystallography. So you can think of the unit cell here, which is just one of these little squares, being analysed in terms of these sets of waves in different directions and of different wavelengths or frequencies, so that these adding up would build the density in the unit cell. Now, you can diagrammatically represent the strength of each of these frequencies in what we call the Fourier transform, so that, say, that this strong dot here would come from this set of waves here. So the strength of the feature here represents the strength of this wave in the density in the unit crystal, and so on for all of these. So this is like, you can think of the Fourier transform, as it were, as a tableau which lays out the strengths and directions of these waves into which you can break the object or the image. In X-ray crystallography, this would be the X-ray diffraction pattern, and you can for X-rays you can record the intensity of that, but you lose the relative phase information between the different diffracted beams. And by that we mean that in the Fourier transform, the wave can have an amplitude, that's how strongly it's oscillating up and down, and it have a position where are the peaks and troughs relative to some fixed origin. And the phase matters, it's very important. It's actually more important, it carries more information than the intensity. You need them both, but the phase information is very important. And to demonstrate that, if we have one kind of vertical wave here and add in a second horizontal wave, you see a pattern starting to build up. We're doing a Fourier synthesis. And then if you put in a third diagonal wave here, depending on whether you have a black fringe at the origin or a white fringe at the origin, you see already, even after 3 waves, you're getting quite different reconstructed images here. So the phase is key because it tells you where the stuff is in the object or in the image. So that's introduction to phases. The other thing is that in the tableau that I laid out, you notice that the finer fringes were laid out in the tableau further out, so that in large radius in the Fourier transform corresponds to finer features in the object or the image. And this is demonstrated here by a very old picture taken from Taylor and Lipson's Optical Transforms book. If you take this duck here and take its Fourier transform, it looks like this sort of blobby thing here. But if you then do the inverse and reconstruct, if you went from this, you'd get back here. But if you cut out the outer finer parts of the transform, there are fine features out here, then you lose the fine detail in the duck. You've kind of lost its beak, you've lost its eye, you've lost its wing. And if you cut it down even more just to the central part, then all you get is just a very vague blobby part. So the further out you go in the transform, the finer the details you put back into the object or image when you do the Fourier synthesis and add the waves back together again. And the last of these kind of picture slides about Fourier transforms is this projection-section relationship, which I mentioned had been exploited by de Rosier and Klug in their Fagetail paper. And what this says is that if you have a three-dimensional specimen, we've got a 3D duck rather than a 2D duck now, and you take different projected two-dimensional images, so it's like you've put your duck in the electron microscope and you're collecting two-dimensional projections of the duck, then you can split these two-dimensional projections down into their two-dimensional Fourier transforms like this. And then, remarkably, these two-dimensional transforms of projections correspond to central sections, the corresponding central sections, through the three-dimensional transform of the three-dimensional duck, if you could have got that directly. You can't, because you've only collected two-dimensional images. However, you can build up the three-dimensional transform here, a section at a time, by including different views of the duck. And when you've filled in the three-dimensional transform, you can do a Fourier synthesis, you add all the waves together, and you recreate a three-dimensional map of the duck. Now, I mentioned that phases are important, and in X-ray crystallography, you don't measure them directly, you record the intensities of the diffraction patterns, and you lose the phase information. And you have to recreate that by isomorphous replacement. You modify the protein by putting on heavy atoms. This changes the intensities in such a way that if you have different heavy atom derivatives, you get different changes in the diffraction pattern. From these intensity changes, you can recreate the relative phases of the diffracted beams. We don't have that problem in microscopy because the microscope recombines the diffracted beams from the objective aperture in the microscope to give you an image. And in this image, not only is the intensity of each Fourier frequency encoded, but the relative phases as well, because we know where things are, as it were, so you know what the phases are. So when you numerically compute a two-dimensional Fourier transform like this, these in mathematical terms are complex numbers. They have an amplitude and a phase. So in microscopy, we don't have a phase problem equivalent to that which occurs in X-ray crystallography. So this was one of the first single particle reconstructions that Linda Amos and I did back in about 1970, a long time ago, of bushy skunk virus. And you can see now that the the negatively stained images really do contain quite a lot of detail, and using the sort of methods that I've just outlined, finding the orientations, finding positions, combining particle from at least 5 particles, 5 images, we never mind hundreds of thousands now. You see how things have progressed. We calculated 3-dimensional maps of negatively stained viruses at around 20 angstrom, which was nice then. It showed you what the molecular surface properties were of the viruses, and confirmed the architecture of the viruses in the way that had been kind of hypothesized by Casper and Klug for their quasi-equivalence theory for the structure of viruses. So that's basically where things stayed during the first half of the 70s. And what was needed was a different kind of preparation. I mean, this is fine, but all you're doing is imaging the stain coating the particles, so it limits you to looking at the surface details. And the next step forward was taken by Richard Henderson and Nigel Unwin when they studied the purple membrane, which is a patch of a light-driven proton pump in the membrane of Halobium something, can't remember. Halobacter halobium, that's right, I think it's halobium something. And you can isolate these patches, and they preserved them on the grid in glucose. And this provided a stable specimen. It was a good way of putting the membrane patch, which is crystalline, onto the grid. And by taking electron diffraction patterns, you can see that the spots here go out probably to 3 angstrom, something like that. And not here, but in some patterns. So there is extremely good order in these membrane patches. However, what they discovered was that if you study the strength of these intensities as a function of the electron dose that you give the specimen, you find that the intensities fairly quickly decay with electron dose, so that by the time you've even got to 1 electron per square angstrom, there has been significant damage, because electrons are far from a perfect imaging modality, because there are more inelastic, chemically damaging events than elastic scattering, which are the ones that you want for your imaging. So by irradiating the biological object, you're not merely imaging it, you're destroying it. So this is a limitation which comes when you start dealing with unstained specimens, and we'll come back to this. So you have to work at very low electron doses, less than about 1 electron per square angstrom. And when you do that, your image looks like this, which isn't, as you can see, terribly interesting. However, if you take a diffraction pattern or computer Fourier transform of that, you can see there's a set of spots here lying on a regular lattice, which tells you that even though you can't see anything in this, there is actually an underlying order in there, which is revealed by the Fourier transform. So how do we extract useful information from this? And you can, this is just by way of a pseudo illustration of what goes wrong or what goes right. This is photon limited light imaging, but the principle is the same as it would be for electrons. And I've kind of arbitrarily, it's a bit arbitrary this, but I just think it demonstrates the point quite nicely about noisy imaging. I've arbitrarily said that this is 50 by 60 angstroms because this image, where this was taken from, this book by Rose, had 3,000 photons in this area. So that, in this kind of very arbitrary scaling, is equivalent to 1 electron per square angstrom in this context. It's a bit phony, but it gives you a relative scale. So if we use 4 times more, 30 times more, 250 times more, 1,200 or 9,000 times more, you see that the definition in the image gradually improves with the number of photons that you record. You can do the actual thing here because you're not going to destroy the woman with the photons in this case. And you can see that it has 2 effects. One is that you record the relative contrast levels, the density levels, much more reliably as you increase the dose. And you can reliably see finer and finer details as you increase the dose. So the sort of details of the striations in the hair and everything become apparent. So by increasing the dose with which you image, you can recover the fine details and get over this noise limitation of quantum limited imaging. So George set up a nice example for me here where rather than just increasing the dose on one copy of the thing, like happened there for the woman's face, you take in the microscope lots and lots of particle images spread all over your grid, and then average those together, which is the equivalent of increasing the dose on a single particle. And you work on the assumption here that the underlying structure, the underlying particle, has kind of unique structure, so that when you average together different images of that structure, the common features will all superimpose and build up, whereas the noise, this quantum limited noise, will gradually average out. So here's a set of noisy images of, as it were, single particle images. And if you take one of them, 2, 5, you've been to see something emerging, 10, 50, and 1,000. We have a single particle image of Max Perutz, which I think is appropriate for this lecture theatre. And you can see now how by superimposing and averaging, you go from just a very noisy, say, quantum limited image to something where all the fine detail can be seen. So this is the basic principle. So in the case of purple membrane, you can do this averaging by taking out just the crystalline frequencies here, the amplitudes and phases which lie on the regular lattice. And by doing that, you can compute an average projection like this, showing the unit cell of bacteria rhodopsin crystal, and showing that it's got 7 transmembrane helices. So this was really the first good attempt at an electron microscope, at a membrane protein structure, and it was certainly the first time that a protein structure had been kind of solved internally by using electron microscopy. Now, you can see here this kind of ringy pattern. This is what happens when you take a very long exposure, so that essentially you've destroyed the purple membrane, but you make a high-dose recording of an image of the carbon film that's supporting it. And you can see that it's covered in these circular fringes. And these arise because the images are recorded with quite a large degree of defocus. Because there's no metal stain here, it's just biological material, light atoms, you get very little contrast. The specimens are weakly scattering. And therefore, to introduce contrast, you defocus the microscope, which is a bit like putting they're weak-phased objects, these. And as you know, in light microscopy, you can't image weak-phase objects, like transparent things, unless you put in a phase plate. And we don't really have phase plates that are effective for the electron microscope. There are attempts, but they're not very effective. So you can do a crude kind of phase plate by defocusing the microscope. And the first kind of quantitative analysis of this was made by Harold Eriksson here when he was in the lab with Aaron. They worked out kind of mathematically what's going on. What one finds is that these are cross-sections through this kind of a pattern. If you took a cross-section and plotted the density across here, then you'd get this kind of a rippling curve of strong and weak rings. And the place where these rings occur depends on the amount of defocus that you've taken the image with. So this is an image taken exactly in focus. This is at 900 angstroms under focus, so-called Schurter focus, and this is 5,000 angstroms, which is much more typically what microscopists used anyway, because when you're sitting at the microscope and focusing through, the image kind of looks crispest, if it's a negatively stained specimen, at about 5,000 angstroms. And what these represent are the transfer of contrast in the image at different spatial frequencies. So these are high spatial frequencies out here and low spatial frequencies here. And what you can see is that as you go to increasing amounts of under focus, this first peak of contrast here, it's actually shown as a dip, but it's strong contrast, gradually moves into lower frequencies. So this would be 20 angstroms. And therefore, if you were imaging a negatively stained object, this would be a good defocus to work at, because you're giving maximum transfer to frequencies in the range of 20 angstroms. You'll hear a lot more about this. They're called contrast transfer functions, or Ctfs. And as I said, this is going to come up a lot in the course of during the course. Now, because this thing wiggles, it goes negative and positive and negative positive. This means that the spatial frequencies from the image are unfortunately transferred from the object, transferred to the image with alternating contrast. So it might be white contrast here and black contrast here and white contrast. So although you're transmitting information about the object to the image, you're doing it in such a way that different frequency bands get inverted in contrast, and black becomes white and white black. And you can demonstrate this by an optical transfer, by an optical frequency test chart. I'm sorry, this makes me feel ill to look at. I won't keep it up long. It probably makes you feel ill as well. This is from Goodman's book on Fourier optics, which again, if you want to know more about this, is a good source. And if you take this out of focus, you can see that the frequency bands here, as you're coming out, everything's all right. You get a fuzzy area here where not much is going through. And I think you can see that this radial spoke here, this black spoke, is opposite a white spoke up here. So that in this frequency band here, the contrast has been reversed from the initial contrast of black and white. It's become white and black. And then as you go to higher spatial frequencies, which is in here, you can see that it's flipped back again. So the black here is now opposite white, and it's opposite black here. So defocusing is good for introducing contrast in the microscope, but it comes with this side effect of requiring compensation for these phase flips. So Richard and Nigel tilted their purple membrane in the microscope, as they had to do, because you didn't have the helical symmetry that you had in the phage tail. You can collect three-dimensional data and make a three-dimensional map of the specimen, of the protein molecule, showing the 7 transmembrane helices. And they took this on to the stage of essentially having a crystallographically refined map from which they could hypothesize a mechanism for the light-activated transport of protons. So this shows you that, once you get to high quasi-atomic resolution, as in protein crystallography, then you can start saying something interesting about the biological function. And that's why we need to get to high resolution. So that was fine and good for two-dimensional crystals, but lots of specimens don't conveniently come as or can be converted into two-dimensional crystals. And the next preparative breakthrough in electron microscopy was by Jacques Dubochet and his colleagues in Heidelberg in the mid-1980s. Marc Adrien particularly, who sadly died just a few months ago. And they published this article in Nature in 1984, viruses again, and what else? And then a much more extensive and large review in Quarterly Reviews of Biophysics, which is available in the library here. And again, for students, these are 2 papers you should go and look at, go and read them. They're very interesting, and they set the scene for what you're going to do with your specimens. So just briefly, many of you will know this already. You put your specimen onto a grid of some kind, blot it so there's just a very thin film of buffer with the virus or whatever it is in left, and then you freeze it into liquid ethane slush kept at liquid nitrogen temperature. And the film freezes so rapidly that the water can't have time to crystallize into ice, and it just makes a glass-like substance in which your virus particles are embedded. So this is a very kind of friendly environment for the particles. They're in solid water, and you mustn't then let the ice warm up, the grid warm up, because then the glass-like structure will turn to crystalline ice, and then that'll damage your specimen. So you end up with a grid something like this, with the virus particles, or whatever your particles are, suspended over holes in the carbon film and embedded like this in a thin film of water. And from that, you can take images like this. A virus, again, this is the core particle from hepatitis B virus that I've worked on for some years. And this is a picture taken by Bettina Bircher. And you can see now very nice details in the particles, no stain, or everything you see here is biological scattering material. So you have some chance of finding out biological structures, not merely biological envelopes. So just to recur to this business of transfer functions, this is a picture again from Dubochet and his colleagues. This is a virus particle, Semliki forest virus, taken pretty close to focus and then successively more and more under focus. And I think you can see that these, here it's difficult to make out anything. You can see the particle, but the detailing is very fine. But by the time you get out to here, you can see lowish resolution features with very good contrast. And the equivalent of that in terms, if you take a Fourier transform of one of these images, you end up with one of these kind of ring-like structures, and the cross sections through this are the transfer functions that I talked about just now. So that as you go successively more and more under focus, you see that this first peak of the phase contrast transfer function comes into lower and lower spatial frequencies, so that the information is being transmitted from the specimen in your holder to the image on the film or the CCD in a more and more low resolution way, so that you're getting higher contrast. So an image like this is going to be good for finding particles, for finding their orientations, that kind of thing. But it comes with the side effect that the more you under focus, the more you have to deal with these ripples in the transfer function. I mean, there's not a problem. They're dealable with computing. You can just fix them. But it's something that you have to be aware of. And when you look just natively at the image with no processing, you're looking at an image which contains a considerable number of optical artifacts. So the scheme in all these, this is a slightly old-fashioned slide for icosahedral viruses, but it kind of encapsulates what still goes on in something like RelyOn. It's just done in a modern way. So you digitize your micrograph. In this case, it doesn't. It comes flying off the Falcon detector faster than you can cope with it. And you then select particles, you find their centers, you refine centers and find orientations, and you then build yourself a set of particles with known parameters of orientation and position, which you must know in order to put this stuff together correctly. And from that, you can calculate a three-dimensional map. In this case, this is papillomavirus, human wart virus. From this map, you can make calculated projections, which are obviously much cleaner and nicer than the single images, because they've been averaged icosahedrally and from different particles. And then you start introducing more micrographs, more particles, find their centers and orientations, larger sets of particles, better maps with higher resolution, and you cycle round this kind of an iterative procedure. I say it's not done quite like this in RelyOn, but the principles, I think, are somewhat similar. So using those kind of techniques, Bettina and I calculated a map of the core of hepatitis B, which actually has, it's icosahedral, but has 4 independent subunits, blue, red, yellow, and green here, in the asymmetric unit of the icosahedral shell. And we were delighted when we found that each of the 4 density distributions looked here very similar. And we managed to, how do you know how you're getting on? I mean, when you have a map like this, how do you know how good it is? It's nice when you look at it and see the details, and these tubes turn out to be alpha helical structures, so it's all very nice. But how do you know how you're doing? And to do that, one can calculate something called a Fourier shell correlation. And in this, again, we're dependent on Fourier transforms, because if you take two independent sets of particles, and it's important that they are independent sets, and process these through to make two independent maps, you can then take the three-dimensional Fourier transforms of these maps, so you've got frequency representations of your data, and then you can compare spherical shell by spherical shell through these two independent three-dimensional Fourier transforms and plot the correlation coefficient. So that's what's done here, and it's called the Fourier shell correlation. And again, it's something you're going to be hearing a lot about during the next couple of weeks. And here, this shows you what happens as you increase the number of particles. So here we had only 200 particles, so the correlation, the goodness of agreement is falling off fairly quickly with resolution. So we're about 10 angstroms is about here, something like this. For 1,000 particles, it's better. For 3,000, and for nearly 6,500, and then fully icosahedrally averaged here. So you can see that we have a very nice flat correlation here. Everything agrees exactly out to about 10 angstroms, and then things fall off. And you can specify the resolution of your map by various criteria as to where you think this becomes insignificant. I don't want to go into the technical parts of that. Richard will probably do this later on in the week. So this tells you how well you're doing, and tells you whether the map that you finally come up with contains detail, how fine the details are that it contains. And in this case, we had a map at about 7.4 angstroms by a fairly conservative measurement, more like 7 angstroms by a less conservative one. And we managed to do a complete chain trace, assign the sequence here. We actually numbered the chain trace. And when Sam Wynne and Andrew Leslie solved the crystal structure of the same thing a couple of years later, fortunately we found we'd got it right. So this was the first single particle structure where a protein had been solved in terms of a correct numbered chain trace. So that's it was another kind of landmark in 1997. So there things stood for a little while, and it wasn't until several years later, about 2000, that there was another breakthrough. The microscopes had been improving, the stages were getting better, the detectors were getting better, the brightness of the guns were getting better. So there was a gradual improvement. And then in 2010, so quite a gap from the hepatitis B structure, Hongzhu and his colleagues and some other people with a different virus, Nico Grigoriev using rotavirus, essentially had the next breakthrough in resolution, where with a virus called aquariovirus, they could you can tell how good your images are by how coherently these fringes in the transfer function go out in the Fourier transform. It's difficult to see here because it's not a very good reproduction. But basically, they were seeing ripples in the transform here to around 3.6 angstroms. So this told you that if you process these images correctly and did all the corrections that you have to do, then you ought to be able to create a three-dimensional map of aquariovirus at around 3.6 angstroms. And this they did. So it's a multi-shell virus structure. I don't want to go through the details. We're not talking biology today. We're talking kind of landmarks in image processing. This is here is a 72 megadalton virus. So they've actually solved by single particle methods a multi-shelled single particle structure of 72 megadaltons. And I believe this is the largest biological structure that's been solved in this kind of complete atomic detail, and you can see all the protein structures to go to make up the multi-shelled nature of this structure. And there's also this is called an infectious subviral particle because it's already had one coat removed from it, and it's in a prime state ready to infect the cell that it's going to infect. and they could actually detect the cleavage of a critical bond, which was known to be important in the impriming of this for infection of the host. So again, if you get to atomic resolution, you begin to be able to say something interesting about the biology of the system. That was 2010. Several big virus structures came out. They're like rocks, they're easy to orient, and they're high contrast. So as all the way through, it's been viruses at the forefront of technical developments. Now we're here, and I come back to my second slide, I think it was, stolen from Werner, the revolution. And this was in Science just 2 or 3 weeks ago, and I think it's remarkable. So we have the mitochondrial ribosome from this lab, Alexei and his colleagues. We have this transient response channel potential channel from Liao and et al. And we have this nickel ion dehydrogenase from Allegretti, which is Werner's group. And you can see that the size of these things is getting steadily smaller. The bigger something is, the more information you have in a single image of that particle, and therefore the easier it is to orient and find its position. So as at a given level of noise, as the structures get smaller in molecular weight, it becomes harder to reliably determine the orientations. And we're going here from the ribosome subunit was 1.9 megadaltons, this hydrogenate is 1.2 megadaltons, and this ion channel is only 300 kilodaltons. And I think this currently is the smallest published structure from single particles at quasi-atomic resolution. So here they're claiming 3.2, 3.4, 3.36 angstroms. So these kind of density maps are as good as you can get from the equivalent X-ray crystallography if you'd been able to crystallize these structures. And of course, with this, you don't need to crystallize them. You just need to have a nice clean prep in a tube. You put it on your grid, put it in this TARDIS box that we looked at originally, and these wonderful structures come out. So here you can see the base stacking in RNA. Here you see the side chains, the amino acid side chains surrounding the pore hole. And here you see the very nice side chain details on a stretch alpha helix. This is where we are now, as of 3 weeks ago or something, when this was published. And as I said at the beginning, it's an extremely exciting time. And if you can make your specimen, if it's bigger than about a quarter of a megadalton, you can make it cleanly, get it in the microscope, you have every chance of getting an atomic structure out of it. Just to summarize, it's been quite a long journey, a long story. How did we get here? Key things have been the improvements in negative staining. First of all, the negative staining technique, which, it is still useful. I know people now say you just screen in cryo anyway, but I think some of you will sometimes find negative staining still to be useful for some projects. We then had the glucose embedding, which is good for two-dimensional crystals, but somewhat limited to that. If you embed single particles in glucose, you don't get sufficient contrast. And then we had the cryo-embedding for single particles introduced by Dubochet and his colleagues. So these were the kind of 3 key steps over the years for improvements in specimen preparation. For microscopes, well, field emission guns are much brighter and more coherent, so you get better images. Much better stable cold stages. They don't drift nearly as much as they used to. You want to kind of eliminate, obviously, specimen movement while you're recording your image, because if the specimen is moving while you record it, it blurs. That's a problem. And recently, we've got much better electron detectors, which can record faster, and they have better DQE, detective quantum efficiency, and you'll be hearing about that later in the week. It just means that the detector is recording the imaging electrons without adding its own noise to the image. So the noise in the image is just limited by the imaging conditions, not by the recording in the detector. And of course, the processing has improved out of all recognition. When Linda and I did the bushy stunt virus that I showed at the beginning, I think it was probably 2 days processing or something for 5 particles. I don't remember, but it was a long time. You wouldn't be patient enough. So bigger, faster computers are key, and we're probably going to need even bigger and even faster computers. You've got a very good setup here now. Much better algorithms now for treating the noise properly. We were always kind of respected the noise, but we didn't deal with it in proper statistical way. And George's Reliant program now is doing a vastly better job on that. And of course, most recently, here we have the movie processing where you time slice the recording of your image so that if there is movement during the recording, you can actually, if the images are slightly displaced, you have a good chance of realigning the images, and therefore you can, at least to some extent, remove or de-blur the images. So that's been important as well. And George asked me to stick my neck out and look in my crystal ball. I'm not really going to do that. Some of these things are obvious. I think the key thing at the moment, I mean, the improvements have been so stunning that we're almost there. However, there are 2 things that stand in the way, and this is the key one, I think. This means understanding and improving the specimen behavior in the beam. It's quite obvious that when you put the beam on to record the image, the specimen either physically moves or charges or both. And it's that which is really limiting the quality of the images one can record on these current detectors. And Chris Russo may say something about this, I don't know, but his work is going some way to attacking this problem. And I think that's really going to be key for the next jump forward. We're going to have even better detectors. Richard and his colleagues are working on detectors where you essentially do single electron counting. You can't do better than that. And if these 2 things work out, then we'll be able to go to smaller non-symmetric particles, because the noise in the image will be much less, the contrast will be better, the features will be sharper, you'll be able to align them, and so on. We'll be able to look at heterogeneous particle populations. That's already happening to a certain extent, but with better images you'll be able to do a better job on that, whether it's because it's a somewhat flexible particle so that domains move and you have them in different configurations, or because you've got a subunit which is present on some complexes and not others, or you have a ligand or something which is on some complexes and not others. So I think we'll be able to deal with heterogeneous particle populations. I think we still need a bit of work on quality control and validation. It's really important when people publish maps, make claims about them, that we know we can trust it. And various kinds of quality controls are being developed here and elsewhere, and I think that's important. And something which I haven't touched on at all in this talk is cellular tomography. For something where you have repeated copies in your tomogram, then subtomogram averaging is going to be important. But if it's just cellular tomography and you want to see the general structure in the cell, I think that's going to be problematical for a long time, just because of radiation damage. And because it's a single unique structure in a cell, it's very hard to see how one can overcome the radiation damage. So that's it. As I said, for you young guys, this is a really exciting time. It's all happening here. A lot of the development is happening here now. We have the computers, we have the microscope, we have all the technology. Get on with it.
Thank you very much, Tony. Questions for Tony?
Silence them all. They save the technical questions for when you give a real lecture, rather than just farting around like this.
I had a question. Dealing with heterogeneous particle populations, to what extent do you think it will replace biochemical purification? Do you think one should even try?
I wouldn't want to put a cell lysate on my grid. Mind you, we have done that with some of our... Yeah. we have done that with you. I don't know. The better specimen you can make, if you work on the biochemistry, then the better it will be. But Sure, I mean, you can't have too much rubbish around. You've got to be able to recognise your particles. I mean, I think the work on the ribosome, I mean, you and Israel have talked about this, where the imaging was an integral part of improving the biochemical preparation. So I don't think these things should be divorced. You should be imaging your prep as it develops. But you want the best preps, I think, really, yes. Don't, if you can't get really good preps, then see what you can do with less good ones.
Right. Okay. If there are no more questions, then I see you all tomorrow morning, 9:20.